Thank you. So I will try to tell you something about glycans and their role as biomarkers, but also functional effectors in different cardiometabolic diseases. So we all know that we all know that up to 80% of all the smart drugs we are giving, but also 80% of different lifestyle interventions are actually wasted because we are giving them to the wrong people. We are giving them without adequate biomarkers, which would help us to stratify patients into those who will and who will not respond to a given therapy. So once we will have good biomarkers, then we will be able to stratify patients before we start therapy or before we initiate a lifestyle intervention. And there was a huge hope that genomics will resolve this. And hundreds of billions were invested in different genomic testing, but this did not deliver much. And one of the reasons why this did not deliver much is that the majority of diseases are actually not genetic. Only 20 to 30% diseases have genetic background, and majority of others is actually lifestyle. And glycans are a large part of inter-individual variability. Even blood groups are chemically glycans. The reason we are not talking much about it is that people are not looking. And once we look, we see. And one of the good examples is even in this pandemics, when at the beginning of the pandemics, even the director of NIH was writing about the S-glycoprotein without a single glycan on its structure. This is just a polypeptide part of a molecule. Well, actually, S-glycoprotein on its surface is more glycans, so everything hairy here are glycans. And these glycans are not just decorations, they're actively participating in the interaction with the protein receptors. And the majority of proteins are glycosylated. And if you want to see the complete picture, we have to look at the glycans. And here I have to say a word of warning, glycation. So HbA1c, glycated hemoglobin, this is something completely different. Please do not think about what I will be talking in this presentation in any way related to what you know about the glycation HbA1c. HbA1c is a chemical damage caused by increased glucose. Glycosylation is a complex physiological mechanism. When I try to explain glycosylation to people who do not have experience in glycosylation, I use this analogy. So we all know what are genes. Genes are blueprints telling how you can build this Batmobile. And then together with these genes, we have a big instruction book. Instruction books is actually epigenetics. Epigenetics tells us which gene will be active in which cell in which moment, and it's regulating activity of these genes. The genes code for polypeptide. So polypeptide is a basic component of a protein. In a way, this Yugo car is a basic car. It moves. If you look at it, it moves. You can go from point A to point B, but it's very hard to consider this to be a fancy car. Once proteins are modified, and the majority of structural modification are actually glycans, then you get a glycoprotein, which is something like this Rimac-Nevera hypercar, which is one of the fanciest cars in the world. So glycosylation is evolutionary improvement of proteins, which makes them way better than just the polypeptide part. And this is not only me saying that. There is a series of documents, European Science Foundation, there is US National Academies, policy documents, which clearly say glycans are directly involved in the pathophysiology of every major disease. Problem with glycans is that they're chemically very complicated. They're branched, non-linear structures. They're not easy to analyze, especially not in a high throughput manner. So the number of laboratories in the world who can do high throughput glycomics is limited. And we went in that direction nearly 15 years ago. We already analyzed over 150,000 people. but not any people. We analyzed people from the best genotyped, phenotyped, clinical epidemiological cohorts all around the world. So where is a good cohort? I try to contact the people, gather samples, and analyze them. So in addition to glycomic data, we have all the genetic, epigenetic, metabolomic, microbiome, and so on data for the people whom we have analyzed. Something which makes glycans different from other biomarkers or other molecules is that they integrate genetic information, epigenetic information, and environmental factors. So protein is a direct product of a gene. Glycan is a product of dozens or hundreds of genes. So all the polymorphisms in these genes, all the epigenetic regulation, all the direct interactions with metabolites are affecting the final glycan structure, which would be added to a given protein at a given time. And this is tightly regulated. In the past decade, we did a series of genome-wide association studies of the glycom, big part of it of the IgG glycom. And we have learned that there is a network of over 40 genes which works together to glycosylate IgG. Only 5 of these genes, which were our GWAS hits, were actually glycosyl transferases. So the enzymes which add glycans to immunoglobulins. All other genes are some kind of a regulatory genes, which make a decision which kind of glycosylation to put on IgG. And this is protein specific. For example, we compared, we did a parallel GWAS of transferrin and IgG. So the same people, the same samples, we analyzed transferrin IgG glycosylation. Glycan structures are very similar. So both IgG and transferrin have a very similar biantennary glycans. And the enzymes which make them are exactly the same. But the regulatory network for transferrin IgG are completely different. So glycosylation is a regulated process where a number of genes work together to make a decision what kind of glycan to put on a specific glycosylation site. And we know that glycans on IgG are functional effectors which have multiple roles in balancing inflammation. For example, short glycans like this are highly promoting inflammation, while extended glycans with galactose and salic acid are actually suppressing inflammation. Core fucose is blocking ADCC. Glycans without the core fucose are promoting ADCC. And the field of immunology is starting to realize the importance of glycans. They have been ignoring them for decades. And recently they started to appreciate the importance. And for example, our review, which was published in 2018, is still the most downloaded paper in cellular immunology today. So immunologists are starting to learn about glycans. And what we have learned is that the genes that regulate IgG glycosylation, so those GWAS hits which we discovered, are highly pleiotropic with a number of different diseases and conditions. We found 94 different phenotypes, including the expected things like Crohn's disease or multiple sclerosis or lupus, where the same genes are the risk factors for disease and regulate IgG glycosylation, but also unexpected things like a Parkinson, where there is a pleiotropy, HDL, dementia, and so on. So these genes which regulate glycosylation in the same time are risk factor for disease. Of course, this does not mean that the glycans are directly in the pathway to disease, but in some of these diseases, probably they are. Because when we think about alternative glycosylation, so putting a different glycan on a given glycosylation site, we should be thinking that this is in a way functionally analogous to coding mutations. We know that the coding mutation can kill a person. You have a wrong amino acid and you have a non-functional protein, and then you get... can somebody mute themselves, please? I hear noise from somewhere.
Hey, good to see you.
And these coding mutations are a result of a mutation in a single gene. Glycans are product of a network of genes. So these structurally differences caused by alternative glycosylation are not encoded in a single gene, but in a network of genetic polymorphisms, epigenetic regulation, and environmental influences of all these genes. So we have this additional layer of complexity, this network interactions between different genes, which make a decision whether this glycan or this glycan would be put here. These different glycans result in a different structure, in this case of the Fc domain of antibodies, and then directs these antibodies to different receptors and actually have different functional consequences of recognition of the FAB part. And despite the absence of a direct genetic template, our studies on a large cohorts of twins have found that for some glycans, this is up to 70% heritable. Some other glycans are only 30% heritable. So it's mostly environmental, while other glycans are mostly genetically defined. And we did an interesting experiment with a collaborative cross cohort of mice developed by the Grant Morahan in Western Australia. So he started from the 8 founder strains of mice, crossed them for 3 generations, and then inbred them into strains. So we had over 100 strains of mice which were actually cousins. And then when we looked at IgG glycom, some of these strains completely separated from other mice. So only 3 generations of reshuffling genetic polymorphisms, so no new mutation, just reshuffling of existing polymorphisms, created strain of mice which were structurally and functionally different. So actually what we saw a microevolution of immunoglobulins at the level of not mutation, but reshuffling existing alleles. And when we looked at the large cohorts, the strongest factor affecting the IgG glycom composition was age. And initially we saw this as a problem because we could not compare patient cohorts with controls if they were not perfectly age matched. But then we realized that this is actually a very important biomarker of lifestyle because the pace of this change was regulated by lifestyle, not by genetics. And what is shown here are 5 different cohorts, different glycans. Blue are men, red are women, and they all changed in a very similar way through age. And then we developed this glycan age clock of biological aging, which was incidentally published on the very same day as the Steve Horvath's methylation clock. So they were both published on the 10th of December, 2013. And often people ask to compare epigenetic and a glycan clock of aging. So I made a small comparison from my viewpoint, of course. And this is that epigenetic clocks are based on information. So when Steve developed his clock, he selected, I think, 353 or something like that CPG sites. But many other combinations of several hundred CPG sites would give exactly the same molecular clock. So this is information. This is not direct effectors. While glycans, there are molecular effectors of inflammation. We know that these old glycans promote inflammation, young glycans suppress inflammation. Another very important aspect of epigenetic clocks, which people often ignore, is that each commercially available methylation clock is different. They are all based on Steve's clock, but they usually analyze a smaller subset of CPG sites. And we cannot just take conclusions from one of the clocks and extend it to the other one, because we have no idea what is explaining the difference for each of them. And this is a big potential pitfall at the moment, because people are taking different clocks and interpreting them based on the initial Steve's clock. With glycans at the moment, we don't have this problem. There is one glycan clock. And of course, I have to disclose, I am one of the inventors of the clock, and I'm one of the co-founders of the company. So clearly I'm here in a conflict of interest. So please judge the facts and not my interpretation. So this glycan clock is based on 24 individual glycan structures, which are attached to immunoglobulins. Some of them are pro-inflammatory, some of them are suppressing inflammation, and they change with age. We try really hard to substantiate every claim we have with a solid research. So we published so far over 200 papers in this field, some of them in the leading journals, including the Circulation, Gastroenterology, couple of Nature family journals. And I'm really trying to show you the hard science and not act as a representative of a company. So one thing we learned is that people with different diseases in a way resemble aging. So when we look at this figure, the last 4 bars in each of the panels, this is aging. This is the change with aging, with these glycans without galactose going up, these glycans going down, these going up, and so on. And then we compared a number of different diseases, dozens already, maybe 10 or 12 of them are here. And we see that people who are diagnosed with the disease generally have their glycans changed in the same way as we see with aging. And very often, these changes actually happen before the disease is diagnosed. So we believe that at least in some of the cases, these molecules which drive inflammation are actually contributing to disease development. I will show you a few examples. And one maybe the most, the strongest one we have are the cardiovascular diseases. So the first paper on this topic we published 4 years ago when we looked at the 2 large cohorts, one together with Tim Spector and his twins UK, and with Jim Wilson from Edinburgh. We analyzed a couple of thousand people, and we noticed that the IgG glycan composition associates with the cardiovascular disease risk score. And then based on that data, we went to the EPIC cohort. We got samples which were collected nearly 30 years ago. And then there was a clinical follow-up. So we actually knew at individual level who did and who did not develop heart attack or stroke. And in men, glycans were as predictive as entire AHA score. In women, a single glycan plus age was more predictive than the entire AHA score for the future cardiovascular events, either heart attack or stroke at individual level. And currently we did 2 replication cohorts. This is still unpublished, but this is something what we again saw in 2 additional prospective cohorts. This will be published probably later this year. Then we looked again together with Tim at longitudinal samples of the same person. So the same person sampled several times. And we noticed that glycans today predict future hypertension diagnosis. And this was replicated in another cohort in Germany. And there is an independent line of evidence, mostly done by Phil Scholl in a UT Southwestern, but recently replicated by another group, which indicate that these hypocellular immunoglobulins, these old immunoglobulins we are seeing, are actually functionally driving hypertension by binding to the Fc gamma receptor 2B on endothelial cells and either promoting, if there are these young glycans, or preventing the release of nitric oxide and the effect on the cardiovascular system. So we teamed up with Phil, and together we did a large experiment which was published in Circulation 3 years ago, where we have shown that by improving the IgG glycans, we can actually prevent development of hypertension in mice. So the mice were put on a high-fat diet, and mice on high-fat diet became obese, and they developed hypertension, and they lost cellulation of IgG. There is a dietary supplement called MANAC, N-acetylmanoxamine, which was given to those mice, MANAC promotes cellulation of IgG, and the mice on MANAC did not lose IgG cellulation, so their IgG cellulation was at a normal level. They did become obese, but they did not develop hypertension. So in an animal model, improving IgG glycom can actually save obese mice from hypertension, which is potentially extremely interesting for humans. At the moment, the MANAC is too expensive for human trials. It would cost millions just to have a clinical trial with mice, with humans, but I hope one day this will be done. Another very interesting area where glycans are extremely important is diabetes. These are not IgG glycans that much. It is mostly total plasma glycom because it affects half-life of different receptors, including the glucose transporters and the insulin receptor. There's a lot of data from other researchers. What we did, we looked in the large cohorts and we saw there is some information about diabetes in cohorts of healthy and people with diabetes. Of course, diabetes is not a single disease. It's very complicated, so it's not a perfect signal, but there is some signal. And interestingly, there is signal even before people get diagnosed. This was a small study, but it was enough to persuade the Epi cohort to give us samples again for the people who developed diabetes. We had, out of initial 27,000 people, 800 of them developed diabetes during the follow-up. So we analyzed glycans there, and we have shown that the total plasma glycom composition was as predictive as the entire German diabetes risk score. So in the plasma glycom, there is something which predicts future diabetes. And we know this is not only a biomarker, it's a molecule which actually has an important role in glucose metabolism. So we were wondering whether this is a risk factor, whether this is some kind of a genetic predisposition for future diabetes development, or is this an early change on the path to diabetes? And we did a study on 2,000 twins in 3 time points over 20 years. And we have shown pretty clearly that these glycans from health to disease change in a kind of continuous way. So changes which we see in glycans are following the path to diabetes, from health to diabetes. And there seems to be a subtype of diabetes where glycans are extremely predictive. This was done on a cohort of people who developed a hyperglycemia and acute disease, which is known a risk factor for diabetes. Here, glycans were nearly perfectly predictive. So to put this in a general picture, the idea is that we have healthy people and then we have people who are sick, meaning something is not functioning. They ask for help. They feel pain in some of the organs and then they ask for help. Usually, disease is an endpoint. This is beyond the point of no return. And generally, people with diabetes or cardiovascular diseases, they do not get healed. They just get their condition improved. They can function, but they continue to stay chronically ill. Idea is to find these people early, help them with the early reprogramming therapy and return them back to homeostasis, or even better, find them when there's no illness yet, where only some biomarkers which lead to the disease are changing. Like what I have shown you for the glycans in diabetes, they start to change early. If you find them early, then maybe even just a simple preventive lifestyle change or preventive pharmacological intervention can revert this wrong path, and people will never end, or much later will end with the disease, and they will stay healthy. There are many examples where glycans are functionally important. I don't have time to go through it, but there is an excellent review prepared by the 2 colleagues from my team. It's part of an extremely expensive book, but our chapter is freely available at this link, so feel free to download it. We got the right to share it. And the key question which we have today, and I think this is everybody's question, is can we revert the glycan age of aging? Is there something we can do to stop inflammaging and to slow down the aging clock? And we have examples of anti-aging physicians who are very successful in this. For example, we did already several hundred patients together with Joseph Raphael in New York. And generally, the glycan age of his patients improves dramatically once they start on his therapies, whatever he does. We have some examples of people also on this side of Atlantic. You probably all know Tim Spector. This is his glycan age when he did an intervention a little bit less than 2 years ago. He was really successful in improving his glycan age. Another important element where we think there is a huge potential to do something is the menopause, because menopause also associates with extensive changes in IgG glycan. Men generally age linearly, while women have this accelerated aging around the menopause age. And this seems to be strongly driven by hormones. We did a very interesting series of experiments on a cohort generated in US slightly more than 10 years ago, where they had young men and women where their hormones were chemically blocked. So this was a cohort of women. They had chemical menopause induced. Half of them, a little bit less than half of them, had estrogen with an estrogen patch. 21 of them received placebo. It was randomized, so nobody knew what were they actually getting. And what we have observed is that women on placebo, on average, aged by 30%, so approximately 10 or more years in a couple of months when the estrogen production was stopped, which did not happen in the group who received estrogen supplementation. So estrogen is sufficient to drive increased aging. So depletion of estrogen is sufficient to drive increased aging in the perimenopause period. Then we went again to this cohort of twins, which were sampled for 3 times across 15 to 20 years. Because problem with menopause is that if you measure only hormones, they're indicative very late. Because in the perimenopause period, hormones go up and down like crazy. You have to make multiple measurements to see that there is actually decrease in the hormone concentration. Since IgG glycan is under strong regulation of estrogen, and we have shown this on a molecular level, how this actually functions, IgG glycan is actually integrating estrogen concentration in a couple of weeks, actually 4 to 6 weeks probably, because IgG has a very long half-life. So by measuring IgG today, we are measuring estrogen concentration in the last couple of weeks, and we see that this glycan composition is very predictive. And when we look at the pace of change, it's also very predictive, because what we see on a cohort level, there is a slower increase in a premenopause, much faster change in the perimenopause, and then slower again in the menopause period. And when we look at individual level, so usually we have women who would be very good in their glycan age, and then extremely quickly this deteriorates. Usually they start to panic in this period, but this then goes back if there is a hormone supplementation. And this is really a radical change. For example, if we look at individual glycan levels, so these are individual glycans on IgG, G0, G2, and S are the key components of glycan age. This is the level of change in the premenopause, and then it changes to much, much faster change in the perimenopause period. So women who are being tracked and then they see change in perimenopause, this is one of the first indicators that there is something going on with their hormones. Hormones are not the only way to improve your glycan age. Losing weight is extremely efficient. Bariatric surgery, we had patients who improved for 37 glycan years in 6 months. Not everybody improves that much, but many people have really have a drastic change after bariatric surgery. So this is a radical weight loss. kind of normal weight loss also works. These are, again, the same twins over 15 years. Those who were gaining weight were aging much faster than those who were losing weight. But there is no magic diet. There is no single diet which helps everybody. We had 1,000 people on 5 different diets for a year. And on each of the diets, some people got better, some people got worse. We are all different, and we have to find things which work for ourselves. The same goes for exercise. Often people try to sweat out the bad diet. This does not work. If you overtrain, you actually get worse. It has to be optimal ratio of training and relaxation. Microbiome is extremely important. We have shown that if you do a fecal microbiota transplantation, your glycans can improve. And this is something which we already have on the market as this Glycan Age test, for which David Sinclair and I published an editorial 2 years ago saying that they integrate genetic, epigenetic, and environmental factors into a simple index that can be reliably quantified. And the most importantly, they're not only biomarkers, they're functional effectors which promote or suppress inflammation. You have all probably heard for this Glycan Age test. It's easy. You get the box, you collect samples, ship them back, and get the results. And the key idea is that we can actually help people to get motivated to do something, because we all know what is healthy, but this is difficult. It's not easy to change the diet, exercise, stress, sleep, and not all components are equally important for everybody. So using this test, people can see what actually works and what does not work. And these are examples of some real people going up and down and making lifestyle intervention to change. And the idea is that you get an objective insight into the current state of something which we know is related to low-grade chronic inflammation. It's a quantitative tool which you can use to monitor effects of lifestyle interventions. And we are working with a number of clinics worldwide who are now using it to show that their intervention actually works. And people get fast and objective feedback, something you measure and you show, yes, I have changed. And finally, I have to acknowledge that all the research that we do is generously funded by different research grants. We got over 15 million different euros, different EU grants, which enabled us to do all this research. And this was my presentation, and I'm more than happy to take any question.
Thank you very much, Gordan. Thank you very much. My first question is clearly a lot of time has been given to immunoglobulin G as a glycosylation site, but are there like other candidate sites that you want to focus on next, or is immunoglobulin G the main?
So the immunoglobulin G is something which we have already on the market as a test for aging, because this is the test for which we got approval from the European Medicinal Agency to sell it as a non-diagnostic test. We analyzed many other proteins. One extremely interesting is alpha-1 acid glycoprotein, which is highly related to inflammation and which has been studied a lot in the 90s. But the problem is that the moment we want to do something as a diagnostic test, we enter the regulatory arena. So most of the other proteins we still do as a research collaboration. So if somebody has a cohort, we can analyze the haptoglobin, transferrin, fibrinogen, alpha-1 acid glycoprotein, total plasma glycom. Many different analyses can be done. Problem with these proteins is that we have no ideas what each glycan is actually doing. We look at alpha-1 acid glycoprotein. It has 5 glycosylation sites. They change in a disease. But even for the alpha-1 acid glycoprotein, we don't know what the protein does. We know it's acute phase protein. We know that it's, we call it the carrier of drugs, but this is not a biological function for a protein. So we don't know what is the relevance of a change in individual glycan. Well, for IgG, we know that. We know if you put sialic acid on top, it's becoming anti-inflammatory molecule.
Thank you very much. I have other questions. I think Prof Lauc, who is adjacent to you on the screen, has asked questions, but it's via WhatsApp. He sent it in. Is the glycosylation seen in hypertension, seen in both primary and secondary hypertension?
I have to check. I'm not sure.
Yeah, and another question was, does the change in immunoglobulin G glycosylation status in perimenopause parallel changes in FSH glycosylation status seen in etemopause?
I have no ideas that anybody looked for FSH glycosylation in large cohorts. So the biggest...
Pardon me, sorry. Please continue, sorry.
Sorry, I'm in an outpatient clinic at the moment, so it's difficult to ask a question. But I was just wondering whether the additional question I was going to ask was, is a high sensitivity C-reactive protein not as good a reflector of low-grade inflammation and the inflammasome to the more sophisticated glycosylation patterns that you see in some of these proteins?
Yeah, it's very hard to say what is better because we don't have a gold standard. So there is no gold standard of the low-grade chronic inflammation. So personally, I think that there is very high correlation between high sensitivity CRP and IgG glycan composition at the cohort level. So when you take a large cohort of people, these things correlate. How does it work on an individual patient? I don't know at the moment. And I think the key problem is that high sensitivity CRP would be highly sensitive to this, call it the normal CRP. So any acute inflammation will result in a huge increase in CRP, which will then slowly go down. So although this high sensitivity CRP at the cohort level is very informative, I'm not sure that it will be as informative on an individual level. At least I haven't seen long-term studies looking at the CRP level as a kind of indicator of low-grade chronic inflammation. So it does require a lot of research to see what is each of them actually measuring.
Thank you very much. My next question is, are there any andrological changes that can predict glycosylation status as well? For example, let's change the question to, do females beginning HRT early have a different glycanage trajectory than those who don't engage in HRT at all?
We don't know. So of course. So we are extremely interested in tracking at the individual level, women through menopause, starting early HRT, starting very late HRT. I wasn't able to get a good cohort of women with the frequent sampling because all these population cohorts, they have sampling every 5 years, 7 years, and so on. What we would need kind of a monthly sampling of women for a couple of years. And we got a small cohort now from Nottingham where they were sampled regularly for the COVID serology. So now, fortunately for this COVID serology, there were so many people sampled frequently that we'll catch some of the women going through the perimenopause, but it takes time to get those samples. So extremely interesting. Something what I didn't mention in my lecture, I can't put everything in a short lecture. For example, for men, it's also estrogen. So this experiment in men, if you supplement testosterone, it's saving the glycan age. But if you block aromatase, it does not. So it's actually estrogen which is driving the glycan age. So those physicians who are trying to build up testosterone in men and then use aromatase to block this leakage of testosterone to estrogen, we're actually not achieving this not immunosuppressive, but inflammation suppression, which estrogen actually does. So this is a world of warning. If you're optimizing hormone in men, it's not good to block aromatase.
Can I ask, you've showed some very impressive improvements in glycoage in Team Spectre over couple of years, and also one of the Joe Rafaelis patients. Can you expand a little bit on what exactly, with Tim, you probably know what he's done to himself.
Yeah, so Tim is of course working on a microbiome a lot. So he's trying all these horrible food like kimchi or whatever to get all the possible bacteria. And he also took metformin. So, but very small doses of metformin. This is what he did. At least this is what he told me that he did. Maybe he has some secret things he also does. So we are currently trying to get cohorts of people on all type of interventions like rapamycin or metformin. But the problem is that usually things are highly confounded with other things. So once somebody decides to do a lifestyle intervention, they start thinking about what they eat, the stress level, and so on. So we really need placebo-controlled randomized trials to be confident that we have actually identified what works. Because, for example, I have a horror story from my lab, and it relates to statins. So we did a huge cohort looking at the effect of statins and saw very high correlation between statins and glycans. Then we did another huge cohort and we saw exactly the same associations. So we nearly published a paper saying that statins strongly affect glycans. And then we got a properly designed placebo-controlled randomized trial, and the effect of statin was 0. There was no effect of statin. But what we were seeing the condition while people were prescribed statins. So it was actually the underlying disease which changed glycans and not the drugs. So we have to be very careful.
What was direction of the effect? What did you think? Which way was it? Statins were reducing age or?
It was not the glycan age. It was other aspects of glycosylation. Because, glycan age is the ultimate simplification of the glycome. Because glycans are so complicated that the moment you move from the narrowest field of specialists, people don't understand what you are talking about. So for the glycan age as a product, we made 2 levels of simplification. One is these 3 key indexes, which is degalactosylated, agalactosylated, and sialylated, and then the glycan age. And of course, the glycan age is kind of a general index. It's very good because people understand it. But of course, we are lacking a lot of information. And fortunately, the Glycan age as a company, they report the entire glycome composition. So if somebody wants to go deeper and give additional interpretation, data is there. For example, I think, Nicolina, the cardio report is already available, right?
Yeah, it is available. I would need to enable it per partner, but it is available.
Yeah. For example, we know that one of these glycans is the best predictor of cardiovascular events in women. We cannot make this claim as a company because it's not approved by the FDA or EMA or whatever, but there are sufficient number of publications that an educated physician can make an interpretation of this number. Very soon, there will be also this Meno age report, which will give these numbers for the menopause prediction. Again, there will be no claim linked to that report because, again, this is still not a diagnostic test. But healthcare professional can make an interpretation like any other test which is not diagnostic.
Can I ask a question? Dr. Lauc. As somebody that I've been very interested in glycosylation during the course of my career, and you're talking about the simplification of this, the glycosylation of. So you're talking about glycosylation of proteins, of presumably lipids, of all types of molecules. And you're looking at, N-glycosylation, O-glycosylation. So how do you simplify this huge maze of glycosylation that you're looking at? And how do you interpret all these different types of glycosylation in your studies?
So, of course, different studies will have a different parts of the picture taken. So this Glycan age test, which is now available, is just looking at N glycans of IgG. So first we pull out IgG, release the N glycans, and then quantify the N glycans. And then we get 24 individual structures, which have some common features like galactosylation, salylation, and so on. And then based on the information about the changes of these few glycans in thousands of people, then we developed an index, which we call Glycan age. Of course, there is no golden standard of biological age. So nobody can say this is the most accurate index of biological age because we do not know what is biological age. What we know that these changes in glycans are either promoting or suppressing inflammation. We know that they correlate with the future disease development. We know that lifestyle interventions, which are known to be healthy, improve these glycans. And then based on that knowledge, we can make some kind of a conclusion saying, if these glycans, which we integrate in Glycan age, improve, you have done something good to yourself. We cannot say whether in the same time you might have an early stage of cancer. This will maybe detect some other glycans. For example, the PSA glycosylation can predict prostate cancer. We don't see this here. We also don't see many other aspects because we are just focusing on one of them now. In the future, of course, there will be many different tests, but this will take time, decades actually.
Thank you.
Any remaining questions? Thank you very much, Professor Lauc. And Nicolina, I'll send you the recording as well.
It was a pleasure talking to you. Thank you for your time.
Thank you very much.
Thank you very much.
Bye-bye.
Thank you.